Okay, for this week, we're going to go into the operons, but before that, I would like to start off with regulation and transcription, just a brief discussion, and basically how it is done and how it is being measured or quantitative measurement in the lab. So, as you may know, it's actually not that easy to measure regulation of transcription, especially in terms of the rate of the transcription itself. In terms of quantity or abundance, or more or less, okay, how much the gene is being transcribed or being made or being expressed is not that hard. But okay, how it regulates or basically the regulation is not that easy. So first and foremost, I did say about this. Okay, there are control. Okay, at a lot of along the way throughout the central dogma of molecular biology. So over here, as you can see in this diagram, we have control at transcription and also at post-transcription. So if you look at this diagram, okay, you will see that okay, there are differences in terms of the RNA being made, whereby when you have transcriptional control and post-transcriptional control. So we will discuss this in detail, especially in eukaryote. I'm not going to go into detail now because I would like to basically more or less talk about the measurement of transcription rate, which, like I said earlier, is not going to be easy. But it has been done. It could be done. It's just not favorable among scientists. But transcription rate could be measured using this four kind of study. First is pulse labeling assay, nuclear run-on assay, polythene chromosome, and study of nuclear RNA. Alright, so let's have a look at each and every four of them. So pulse labeling study, the procedure is actually, this was done way back in 1980s, but it's still being done now. It's just that the radioactive is being replaced with more safer label. But more or less either you're using radioactive or kind of like a safer label. So the nucleotides are labeled and of course added to the cells, as you can see over here. So after 5 to 10 minutes of incubation, this was done in room temperature. So the cells were harvested and all the RNA were isolated. So the labeled RNA will then be hybridized to dot blood to contain DNA from genes where the transcription is being measured. So how is the measurement? There is a calculation and a formula for that. So synthesis of this RNA could be measured by adding radioactive ribonucleotide, as we have seen. So this is done by ruidine labeled with tritium, different kinds of radioactive, and measuring the level of incorporated radioactive, the level of radioactive present, and also the duration of it being active. So the pulse labeling minimizes degradation of newly synthesized RNA by exposing the cells briefly to label uridine. And there are direct ways or indirect ways, but most of the time, direct ways of measuring transcription rates is actually being done using pulse labeling. The indirect is less being used. I don't think I put any one of the example. All the four is actually direct way of measuring transcription rate, and of course transcriptional control. So as you can see with the pulse labeling study, there is a limited application because it requires very short labeling times to minimize degradation. And of course, the short time pulse is most of the time insufficient to measure rates of transcription in a lot of species, especially in eukaryote. Alright, the second method is nuclear run-on assay. So the procedure is actually quite simple. So nuclei is isolated from the cells, and of course you can add it with radioactive labeled nucleotide. You can add radioactive or a safer label nowadays. So after 1 to 2 hour incubation, this is done at 30 to 37 degree. RNA will be isolated from the nuclei, and of course including the label RNA produced by transcribing polymerase running on to the end of the gene. So the label RNA will be hybridized to dot lot, similar procedure, containing DNA from genes where the transcription rate is being measured. So more or less this is how the nuclear run-on assay is being done. Now, most of the time, as you can see from this diagram, this assay is basically based on fundamental that a lot of precursor ribonucleotides are present in cytoplasm, and we know that is true because without which transcription and translation would occur. So by removing the cytoplasm, you will isolate just the nuclei. And of course, at the same time, you also remove a lot of the unlabeled ribonucleotide. So what happens is that RNA polymerase will then stop transcription because there are lack of ribonucleotide. They actually couldn't continue on with the transcription, but it will remain associated with the DNA. So, if label ribonucleotide is added, RNA polymerase will resume transcription to produce RNA transcript. But okay, if not, then okay, it will basically stop, and then you can basically start measuring. So, it could be detected by hybridization similar dot blood assay to corresponding DNA. And this allows quantification of the transcription of genes as well. Now, the third method is basically on polyethylene chromosome. So, what are polyethylene chromosome? Google it up. This is a masterclass. Not everything will be given to you. You need to find some of the information on your own as well. So, polyethylene chromosome, this kind of study is able to directly visualize increased level of transcription. Hence, the keyword over here is visualize. So, idea of the visualization came from salivary glands of Drosophila. Hope you guys know what Drosophila is, okay? So, upon several amplification, PCR, of the chromosomal DNA, it will produce polyethylene chromosomes. So, what happens is that how can we visualize it? Okay, it will exhibit puff effect. Puff means that it kind of like puffy, okay, puffy effect, okay, where decondensed DNA area results in chromosomal expansion. There is, in the next slide, okay, there is I think a diagram or a picture of it. So the puff will represent sites of intense transcriptional activity, and these sites are suggested to contain gene encoding protein due to production of the new protein. Okay, so this is what happens. So transcriptionally active puff, okay, as you can see, this is over here as compared to here. So this is in response to treatment with steroid hormone, ecdysone, and it's not present before the hormone treatment. So this actually, okay, this is kind of like just visualization, not measurement. And the last one is actually the study of nuclear RNA. So basically, okay, we have snRNA or small nuclear RNA. So it is involved in RNA processing reactions, and it has served as model system to study how transcriptional machineries use factors and activators to regulate transcription. And this we will discuss in detail, and there are a lot of things to discuss in eukaryote. So this is mostly used, okay, through transportation across membranes from nucleus to cytoplasm, either again using transcription or post-transcription. So this is the study of nuclear RNA, and this is not a very simple study. For every different organism, whether you prokaryote or eukaryote, okay, you get different results, okay, because how it cross the membrane is basically depends. So as we know that, okay, cells have to cross, so it's not that you can cross it very easily. It's kind of like when you enter into another country, you have to pass through the border control. So this is the same thing, okay, with the cross membrane of the nuclear RNA. So that means that I will just explain it simply. If you want to know into details, okay, you can read a journal, or you can basically just YouTube, okay, there are a lot of explanation on this. But okay, simply said, okay, for anything to enter in from cytoplasm to enter into the nucleus, okay, so we refer to it as a cargo. So it have to have an importer, okay, a protein that basically, okay, will direct, okay, it across the membrane. So it has to have a permission over here, okay. So when the RAN-GTP, okay, basically has given permission, so the cargo is being released, and the importer will also be released. So once the cargo has been released, okay, or wherever it has to go, or whatever that it has to do in the nucleus, so the importer, okay, and the RAN-GTP, okay, will basically bind together, and it will return back, okay, to another portal, okay, to go back into the cytoplasm. Okay. And okay, what happens, okay, is that this is when you want to import from cytoplasm to the nucleus. When you want to basically, okay, export, okay, from here to here, okay, basically it's another story. So one is called import, one is called export. If you know the difference between import and export, you will know why they are named as such. Okay, so I'm not going to explain into detail. Now, if you have to export something from the nucleus into the cytoplasm, okay, similar procedure. But okay, in the nucleus, okay, you have to have, okay, your cargo, apart from being bound, okay, to the exporter, okay, it have to be accompanied, okay, or bound to RAN-GTP as well. So once, okay, all these three, okay, have bound together, okay, it will cross the portal to go into the cytoplasm, whereby the cargo will be released, the RAN-GTP, okay, will also be released, okay, and then you will have, okay, your exporter, which basically will go back here, okay, and then be reused back, and so forth. So as you can see here, okay, over here you have RAN GTP, but okay, when the RAN GTP basically is being released here, okay, it is called RAN GDP, and when it goes back into the nucleus, okay, it will be a RAN GTP back. Okay, so it's I would just explain it as simple as that. The mechanism is actually very cool, okay, but kind of like tedious to explain in like less than 20 minutes. Alright, so I would say I would go towards the end of this. Alright, so the last part is on regulatory RNAs. As you can see, I skipped very fast just now, so actually I want to do another activity, but seeing that probably will not have that much time this week, and I will explain later when we meet. I will skip that. So the last part of this video is on regulatory RNAs. So do you know that there are a lot of RNAs available apart from the mRNA and the snRNA that I was telling about? So we have long RNA molecules, more than 200 bases, and also small RNA molecules, about 20 to 30 bases, which of course they play a role in inhibiting protein coding genes expression. So how do they do this? They basically interact with specific gene or mRNAs to do their function, okay, or to perform a biological processes. So this protein coding DNA are only about 1% of the human genome. Okay, where we know that 3/4 of human genome are basically transcribed into RNA, not that much actually. So these regulatory RNAs would regulate gene expression both at transcription and post-transcription. So it would involve three major classes of small RNA, miRNA or microRNAs, siRNAs, small interfering RNAs, and piRNAs or PE interacting RNAs. I'm not making all this name up, okay, it is as such. Now, gene expression could be inhibited by regulatory RNAs via induction of mRNA degradation, blocking of mRNA translation, or inhibiting transcription. So, okay, for each and every one of these three RNA types, okay, they basically have different length. They are either single or double, and their distribution varies. So they basically require another protein for maturation to make sure that they could perform their function later on. And whether they are found in all species or not, it basically depends. But okay, most of the time with these three are only found in eukaryote. mRNA and siRNA actually are found in certain prokaryotes as well. So the function, mRNA to control cellular gene expression, siRNA to control viral and cellular gene expression, piRNA or PU interacting to inhibit transposons and control our cellular gene expression. Now let's have a look at each and every one of them. So for the microRNAs, they were first discovered bound to mRNA producing gene in nematodes. So binding can occur either in full or partial complementarity in sequences. So single microRNA can bind to multiple mRNAs of different target sequence, as you can see over here. Okay, one single, but it could bind to different mRNAs. So when it binds, it inhibits expression of many different target genes. So that means that it could not produce protein later on, or most of the time the gene will not work properly. Now, second one is the small interfering RNA. So processed from double-stranded RNA as defense mechanism against viruses. So this is mostly found in viruses. So they formed when dicer protein cuts double-stranded RNA, as you can see over here, into smaller molecules, separating into single-stranded RNA. So siRNA binds to the identical sequence of original double-stranded RNA to promote degradation, hence inhibiting viral gene expression post-transcriptionally. This is especially important, for example, probably in whatever that we are having now, COVID-19. So it is used widely to silence genes. So again, this is how it works as well. So you can read on the description. And the last one, and I think this is the last slide. So we have piRNAs or PU interacting RNAs. So it's actually quite small, about 21 to 31 bases. So originally identified in Drosophila germ cells, and the process is basically similar to both micro and also small interacting RNAs, but shortened at both 5' and 3' end. And this doesn't involve the dicer protein. They basically are shortened during the transcription process. How it is still being investigated actually, but it doesn't really involve the dicer protein. So it is found in a wide range of animals with a role to maintain genome integrity to ensure intact transmission to the next generation. So its function basically inhibit the activity of DNA transposons that move around the genome by binding to transposon RNA transcript to induce degradation. So there we have it. So this is basically just the general stuff that I'm going to talk about. So before we move on to the operon, and the first operon that we're going to cover this week is leg operon.